Well, welcome to the Science Cafe. This is the 7th year. For a minute, I wasn't sure. I think so. So we're thrilled to have another great lineup of speakers. We also have the cafe conversations that go on in between. I'm going to tell you these now just in case we get too wrapped up in the topic later on. The next Science Cafe is Ryan Folk out of Geography, and he's going to talk about climate models. And next week, we're going to have a cafe conversation, and that's Pascal Young from Music, African music, and a big bowl of salad. Sounds interesting? Y'all should come. There's going to be drums and dancing. All right. Hang on a minute. All right, so I was told that I needed to show you guys the prizes for adding to the discussion, right? So the deal is, we have T-shirts. This is the Cafe Conversation T-shirt. These are available for tonight. Then next week, if you come back, you could get a Cafe Conversation T-shirt as well. Okay? Now, the deal is, the idea here is if you're asked questions and participate, you get a T-shirt, at least until we run out for today. But there'll be more every time we have a cafe conversation. Does that seem fair? Works out all right. Okay. One other thing you need to know is Sigma Xi. I guess I should introduce myself since maybe some of y'all don't know me. I'm Sarah Wyatt. I am a professor in plant biology. I'm also the vice president of Sigma Xi, which is the International Society for Research, Honor Society for Research. We have a local chapter here, which I'm the vice president of, and they are one of the sponsors of the Science Cafe. The other sponsor is the vice president for Research and Creativity here on campus. He foots the bill. So you want to thank him. Okay, if you see him, thank him. If you don't see him, shoot him an email. One of the activities that Sigma Xi is going to do this year is next Tuesday at 5 o'clock, we're going to have an ice cream social. So if you're interested in Sigma Xi, if you're a member of Sigma Xi, certainly, if you have students or if you're a student looking to be a prospective member of Sigma Xi, come on out. We're going to be in Emeritus Park. You guys know where that is? That's the cool little park that's just right down there, okay? Go out the escalators and hang a left. So we're going to have some ice cream and just a nice social time for a fall Sigma Xi get-together, all right? Now, with all of that, now it's time for the presentation of the week. This is on stem cells. Dr. Monica Burdick.
So we'll do yep, that mic works. Okay, we're good. Okay, so thank you all for coming out today. I feel this is better than having no people here. This is great. So thank you all very much for coming out. And I'm kind of hoping you all will ask me some questions all along because I don't think I have 45 minutes of lecture prepared. So please ask questions. I do want to thank Sigma Xi and Dr. Wyatt for the invitation, the Office of the VP for Research, and Dr. Malé Brun, who's done tons of things for me in my lab throughout the years. And I also, because I see a bunch of them here, I want my students to stand up and wave to the crowd because I couldn't do any of this without them. Okay, not all of them are here, but a good number of them are here. So thank you to everyone. And I do want to thank, again, you all for showing up. Now, I know that Dr. Wyatt said you can send an email off to thank the Office of the VP for Research, but would you like to help me thank them right now? Because I heard that we have the best fans in the MAC. Is this true? Yeah. Okay, that was kind of wimpy. Come on. Show us that we have the best fans in the MAC, not just for athletics, but for academics and research. Come on, let's do this thing. Okay, so will you help me by by following a flow chart of a wonderful cheer. So we're going to start. We've warmed up. This is good, right? Let's hear Science Cafe. Okay, one more. Science Cafe. Big one. Science Cafe. And woo! And we're done. Okay. So my engineers, this is all for you. And those of you who are in Chemi 1800, tomorrow in class, I do expect you to do well on your flowcharts, because we practiced. Okay, so thanks for playing along. All right, so yes, we are here at the Science Cafe. We've all confirmed it. Please forgive my notes. I don't have PowerPoint and it's scary, so I need something to help me out. Now, you may be wondering, what is this engineer doing? Giving a talk about stem cells. Yes, I know. Okay, so first of all, I must reiterate, I'm an engineer. I'm a chemical engineer who happens to look at biological systems, biomedical systems, as my system of choice rather than a classical chemical reactor that you might think of when you hear chemical engineer. I am not a medical doctor. I am not an MD. I am not prepared to answer your questions about medical diseases and treatments in anything other than a scientific sense. So please do not take any of this as real medical advice. I'm not qualified to give that. What I do in the lab, I consider myself a cell adhesion researcher who looks at how cells adhere under blood flow conditions. And as a chemical engineer, I'm thinking about the kinetics of how this happens, how little molecules recognize each other. And because we're looking at a blood flow system, the forces that are involved in regulating some of these cellular recognition events. And while I do think of myself as, in general, chemical engineer who does cell adhesion research, I do some of my systems work under the cancer stem cell domain. I used to do some of my research in the hematopoietic stem cell domain, and we'll talk about both of those types of stem cells today. And I hope some of you will ask me some questions about them. Hopefully questions I can answer too. That would be nice, but that's okay if I can't answer them. I'll try to direct you to places where you can get reliable information, because there's a lot of unreliable information out there, as some of you may know. So why does this topic area interest me? Well, stem cells are kind of cool and interesting. We hear about how they could save the world, cure diabetes, cure heart disease, cure cancer, cure Parkinson's disease, and the list goes on and on. So that is in itself very fascinating. But as an engineer, I would like to understand, how can we do something with stem cells? Yes, as an engineer looking at how things move around in the body, sure, that's interesting because I can apply a lot of engineering models, but that part of me that wants to do something, that wants to, as we say in the Rust College, create for good, must come about by using engineering models and that let's create for good mentality. Now, as an engineer, I'm always thinking, and my engineering students, of course, are always thinking about the costs, the risks of getting involved or modeling or creating any system. So as we think about, in the cancer world, how to target cancer stem cells, we're thinking about how to target them safely. In the therapeutics world, embryonic stem cells, hematopoietic stem cells, how to use them in a way that's going to hopefully treat or even maybe someday cure those diseases, but without doing harm. So there's a lot of ethical dilemmas that we have to think about. Is this cure worth the cost or the risk involved in developing it, in producing it, and so on and so forth? And going back to engineering as a profession, we, as professionals, are guided by a code of ethics. For those of you who haven't gone through any engineering courses or aren't engineers, we are professionals, literally. Our professional representation bodies, for instance, the American Institute of Chemical Engineers, the American Society of Mechanical Engineers, we all have a governing code of ethics. And while those words may be a little bit different, ultimately, we as engineers hold paramount the safety of the community, the public that we serve. So for me, the stem cell world takes into account all of these interesting scientific and engineering models that I find fascinating, but as an engineer, this professional responsibility to do good things without doing harm is something that's always present in all of the research that I do and all of the research that my students do. So the talk that I'm supposed to give today, I think, is stem cells and research and hype. Oh my. So this is actually a reference to the Wizard of Oz. It's not the George Takei, oh my, oh my. I think I lost an and in my title, but that's what that's about. So these things might sound scary or intimidating. Stem cells, ooh, research, ooh, and hype, what? So the Wizard of Oz reference kind of seems appropriate. And then when you think about people, Dorothy, taking the yellow brick road, all in the hopes of going home, for many folks out in, well, actually us, out there thinking, well, stem cells are going to take us down this road to a cure for, as I said, Parkinson's disease, diabetes, heart disease, et cetera, someday. What we're afraid of, I think, as a whole, as a society, that when we pull back the curtain, the wizards behind there are not going to be kind and benevolent people who have society's best interests at heart. They just might be a bunch of con artists. And unfortunately, we hear just as often about the con artists in the stem cell world as the good people, the scientists, the engineers, who are going about and performing their research in an ethical and driven manner. And when I say con artists, I don't necessarily mean, haha, an evildoer who's out to definitely steal your money. I also mean those accidental con artists, people who through a bad decision, maybe making a careless mistake in supervising research or performing research, has ultimately ended up tricking the world, and therefore betraying the trust that the public has given to us through their taxpayer dollars, for instance, or just the fact that they're counting on us to do good things to help cure society's ills or treat society's ills. When we violate the public's trust, we really do harm. What if the entire stem cell research world gets sunk by a series of very bad things, which I'm going to talk about a few of those really bad things that have happened in very recent history. So, with that very long prelude, I will tell you a little bit about my research involving those cancer stem cells, and then take you through some of the things that have happened in the world of stem cell therapeutics. So, I might need a little whiteboard here, so there we go. Yeah, so no PowerPoint, but you can't leave me hanging here. I gotta have something to illustrate some stuff. Whoa. Okay, so we'll do the best we can here. All right. All right, so I'll try to tilt the board here every so often so that everybody can see it. Oh, okay. Whoa.
Whoa, and drop. Let's try that again.
Yeah. Okay, so as I said, I'm interested in cell adhesion research. How do cells move throughout the body? In this current position, I'm interested in how cancer cells move throughout the body. Now, it's thought that they get from the primary cancer, so where the tumor originates, through the bloodstream, then through a series of adhesive events, begin to transmigrate into that organ and form the metastatic colony. And it's that secondary metastatic colony which is particularly troublesome, since cancers can often be treated if they're found in the primary stage with relatively high 5-year survival rates. If the cancer has metastasized, then the survival rate drops precipitously. So we're at breast cancer, if you look at that type of cancer, which we all know is very devastating and very common. If the cancer is found in the primary site, then the survival rate for 5 years is almost 100%. But if the cancer has metastasized, then we're looking at more like 20%. So understanding how these cancer cells move around the body and figuring out the best ways to keep it from happening in the first place, if it happens, from getting worse, is a very important area of research. So, all right, so we've got our cancer cell. It's broken free from the primary site, flowing throughout the bloodstream. And while it's moving around, it's being subjected to a lot of biochemical stressors as well as biophysical stressors. So if you happen to go to Dr. T's Science Cafe last year, and I thought I saw Dr. T's around here somewhere, oh yeah, my colleague, looks at the biophysical stressors that might affect how these cancer cells survive and get into the metastatic domain. And my part, as well as with Dr. Fabian Venencia from Biomedical Sciences, is to look at those biochemical cues. And part of that is thinking about how this cancer cell, we'll just label it CA for cancer, gets down onto the cells that line the blood vessel wall. And using those molecules that allow it to hook onto the lining of the blood vessel wall, let's say this is going to the liver, how it can keep stuck on that wall lining the blood vessels through a series of rolling interactions, ultimately coming to a stop, which we would call a firm adhesion event, and then transmigrating through the endothelium, the lining of the blood vessel wall, to form cancer growth, that deadly thing that is what's so hard to overcome in our anti-cancer therapeutics today. So we want to know, what are these molecules that encourage the cancer cell, as it's moving in the blood flow, to grab on to those cells lining the blood vessel wall, and they have to not just have that biochemical recognition, they need to be biophysically resistant to the blood flow trying to rip the cell away from the lining. And those molecules that maintain the contact and allow that cell to roll along the blood vessel, keep it anchored, and then form that metastatic colony. So there's an awful lot of steps that a cancer cell has to successfully complete for a metastasis to happen. It's not really a very efficient event, but you know, if you have cancer, if you know somebody who has cancer, you're not really thinking about, well, that's really inefficient. We shouldn't care about it. the fact that a metastasis is formed is very troubling and something that we do want to stop if we are ever to fully cure this horrible disease of cancer, no matter what cancer you happen to look at. So that's generally what we look at in the lab, and hopefully I can show you a little video where we have some dark cells in the background. Those are the endothelial cells that line the blood vessel wall. And these white cells, and I know it's a little hard to see in here, the white cells are colon cancer cells that have been recruited from this blurry flow stream onto the endothelium, and some of them are rolling, some of them happen to be firmly adhering. We don't have the right biochemical stimuli in our system in order to force the cells to transmigrate or move through the endothelium, but we do have a tool set in our lab to get the cells to roll, to monitor how many of those cells roll, and at what velocity do they roll. And so, as I said in my introduction, there's a lot of things that go into it. There are some kinetic models that we think about relative to that recognition, as well as the force that's applied on the bond as the cancer cell rolls along the endothelial cell surface. Oops, sorry, lost my train of thought. Ah, okay. So for those of you who know me, I sometimes tend to babble on and then I think something and then I get lost, and so sorry, I think that just happened right now. So I also have another set of videos that hopefully will show up, but if you want to have a closer look, I have some of my students here with tablets so you can see it a little bit better. The cancer stem cell aspect of all of this. So this interaction could happen with all cells in the bloodstream, maybe only a select number of cells. We're not really sure. So one of the things that my lab is trying to do is figure out, is there a preference for a certain subset of cells, for instance, maybe a cancer stem cell or a stem cell-like cancer cell, to undertake this attachment and rolling event. And the way we do this is by using some fluorescent dyes and molecules that recognize supposed markers of the cancer stem cell. Now, why we care so much about the cancer stem cell is that, as far as we can tell, they're very resistant to traditional radiotherapies and traditional chemotherapies that are used to, well, treat your loved ones should they be diagnosed with cancer. So you've got these nasty little cells that can't be killed by our traditional therapies. That's obviously very problematic. And not just survive beyond this initial treatment phase, they sort of hang out dormant, waiting for some other biochemical or biophysical cue to turn on. And when they get turned on, they start making that new tumor. So it's thought that they're very tumorigenic, and they may be metastasis initiating. So while they're hanging out dormant after that treatment, just waiting. Very deadly. Yes?
So you just lost me. When you said, so every cell that rolls along
Uh-huh.
it permeates,
Yes.
does that become the cancer stem cell?
Yes, so that's actually one of the hypotheses.
Repeat the question.
Oh, repeat the question. Oh, sorry. So the question was, these rolling cells, or these cells that are getting onto the endothelium, once they get in there, do they turn into a stem cell? I think I did that question right. So that's one of the major questions in cancer stem cell research right now. When does that stem cell mechanism get turned on? Is it somewhere before the cell enters the bloodstream? Is it sometime within the bloodstream? Because if you're going to survive that transit through this very chemically harsh, very biophysically harsh system, you must have some survival techniques against that, cancer cells. So are the stem cell properties acquired in the bloodstream? Are they acquired while they're touching these endothelial cells lining the blood vessel wall? Or when they're in this tissue space, let's say the liver, if that's where that colony is going to get established, is that where the biophysical and biochemical stimuli are to turn on the stem cell properties? And that is actually what at least one team of researchers believes very strongly, that once they're in this tissue space, actually not just one, several researchers in a camp believe that it's in this space right here that we have this subset of cells that can become stem-like, and in acquiring those stem cell-like properties, hang out in that metastatic space, lying there dormant, waiting for that ultimate signal to turn on, and then we have a very multicellular mass that is the mass that's visualized when the secondary tumor is found. So yes, that's exactly. Now, other people do have other hypotheses, but the one that we just talked about is one of the leading ones. And that is one of the things that my lab is trying to figure out. We have a high amount of curiosity as to whether or not those stem-like cells, regardless of where they attain their stem cell properties, in the primary site or while transiting through the bloodstream, do they have advantages in sticking to the blood vessel wall? Or it doesn't matter what kind of cancer cell you are. Once you get inside that tissue space, that's where the signal comes on to be a cancer stem cell. Yes?
So what makes the cancer stem cell different from normal stem cells?
Yeah. So aside from the, sorry, I keep forgetting that. So the question was, what makes the stem cell different, the cancer stem cell different than the other cells? So when you are diagnosed with cancer, it is not just, well, all exactly the same cells in the tumor growth. There are hundreds of different types of cells, maybe even thousands of different types of cells within that tumor mass. The cancer stem cells are believed to be drug resistant and radiotherapy resistant, which makes them super nasty. They're also thought to be cells that can generate new tumors. So maybe that metastatic unit in that distant site may be entirely attributable to a single cancer stem cell. And they have these properties that I'll talk about with our therapeutic applications of stem cells. But cancer stem cells have this property of being able to make more of itself, so more cancer stem cells that are just as resistant to therapies as the original cell. And these cancer stem cells can differentiate and become those thousands and thousands of different types of cells that make a tumor. So they've got this property of self-renewal and differentiation, which really, whether you're a cancer stem cell, an embryonic stem cell, an induced pluripotent stem cell, you must have if you are going to be a stem cell. So cancer stem cells, yes?
I think, if I understand right, was your question, what's the difference between a cancer stem cell and other types of stem cells? Because that's something that I don't really know, and I'm curious.
Yes, we're gonna get to that one. So the question was, what's the difference between these cancer stem cells and these other types of cells that we hear about? Embryonic stem cells, hematopoietic stem cells, adult stem cells. Yes, very good question. I will totally address that one because those cells are the ones that we usually hear about in the news. Cancer stem cells, unless you're in cancer research, you've probably never heard of them. So how many people watched the Stand Up to Cancer event on Friday? Oh, okay, just a couple. Okay, so I was curious to see, would cancer stem cells be mentioned? Because for me and a lot of other researchers in the cancer domain, This is the thing that you need to talk about if you want to take your research or want your research taken seriously. Now, I overstate that, obviously, but cancer stem cells are a very important part of the research that's going on right now. But in Stand Up to Cancer, I think I heard cancer stem cell said once. So the general public isn't terribly aware of this really nasty stem cell. But we're working on it. I think once we actually can figure out what that cancer stem cell is, because that's kind of a problem for us right now, then we can really target them and figure out how are we going to solve this problem. So I kind of got into this other problem of the cancer stem cells. So we say cancer stem cells, and there's this somewhat nebulous definition. So they're undifferentiated cancer cells that don't have specific duties in the tumor except to make more of itself and to become the other types of cells in the tumor. The molecular definition, so when we type cells, we do it oftentimes by what molecule do you express? Do you express this one, this one, this one, this one? Yes.
Is it just the cancer stem cell that's resistant, or are all the other cells that make up the tumor resistant as well?
Yes. So the cancer stem, repeat the question. Okay, so is it the cancer stem cell that's the only type that's resistant, or are there other types that are resistant as well? The cancer stem cell is believed to be resistant, but many other cells have drug-resistant capabilities to essentially pump out drugs that might be used to target that cancer. They might lack the specific receptors that are the specific molecules that are needed in order to get the therapy to work. So it's not just the cancer stem cells that we're worried about targeting. It's other types of resistant cells. So yes. This one here first.
When did it start becoming resistant?
Okay. So I'm actually, so the question was, when does the cancer start becoming resistant? So is this the tumor as a whole, or perhaps the cancer stem cell?
The cancer stem cell.
Okay, so the cancer stem cell, it's thought that this might be an inherent property of the cancer stem cell to be resistant to these different therapies, largely because these cancer stem cells have a slower division rate of cell division than other types of cells. So if the cancer stem cell isn't dividing as well, yet these radiotherapies and chemotherapies are targeting cell division pathways or pathways somehow related to cell division, yeah, not going to die. Yes.
So that's the drug resistance, so like pumping up the drug out. This kind of mechanism should be regulated by the genes. How can this gene be turned on? All these genes have their two genes are similar copies in the normal genome of the cell.
So,
They just mutated from the cancer stem cell.
so the question is whether or not the cancer stem cell possesses.
This kind of drug with resistant genes should have the similar copies in the normal in the health genome.
Right, so these genes may be present in the cancer stem cell,
Also in the normal cell.
and they may be present in the normal cell. Yep, so this is another issue too. So genes may be present in the cancer throughout all of the cells, but the genes may not be expressed as proteins by the cancer stem cell or really any other cell. Kind of answer your question?
No, I mean the genes to have the function of the drug resistance, they come from other source or just they previously presented in the genome just because due to they become cancer cells with these genes.
So I'm actually not sure of the answer. Okay, so the question being whether or not the gene is active in the cancers.
I mean, you want to know the origins of where is the genes come from. From
The genome, yes.
Of
Uh-huh.
other source.
So the origin of the gene that gives it the drug resistant or other property. Yeah, I don't know if I can answer that question because I, not being an expert in genetics, epigenetic regulation, I probably would just be guessing, and that's probably not good. Also, not an MD. I don't want to pass on information that may be incorrect. So I'm sorry, but we can discuss later. So I think this one first.
Yeah, so I'm just a little bit confused as to what point, when the cell is going along in the bloodstream, what point does it actually become like cancerous and harmful for you? Is it only after it goes like and advances and then becomes a stem cell? Or do you still have consider to have cancer that's curable before that point?
Yes. So this is a very tricky question. The question is, as we have cancer cells in the bloodstream, do we consider this a dangerous condition, a cancerous condition, and or curable? Okay, so this is a very tricky question. While the medical community, medical professionals may or may not be able to see those circulating tumor cells, the fact that they're present is definitely a bad sign. So that means you have cells that have the potential. Now they may not, but they may not establish a metastatic colony, but they have the potential to establish a metastatic colony. Now, sorry, I'll get to yours in a second. The issue of finding circulating tumor cells, being able to count and being able to study those circulating tumor cells is one of the biggest areas of bioengineering right now. So there's this device called Cell Search that's been on the market for quite a while. It is an FDA-approved I'm not sure if it's a diagnostic or a prognostic. I have to be careful there. That essentially tries to capture the cells from a blood sample from a patient and count them. And if you have this number of circulating tumor cells in your blood per milliliter, then something, something, something. And I have to be careful. I don't have the something, something, something in my head right now, but it is clinically relevant. So this is an FDA-approved tool. Right now, there's what's called by the American Association for Cancer Research and the Stand Up to Cancer organization, a dream team that's job really is to study these circulating tumor cells and develop new diagnostics and evaluate diagnostics to see whether or not there is any true clinical relevance across, name your favorite cancer. Are there gene signatures, perhaps, that are associated with these circulating tumor cells? Are there protein signatures? And the list goes on and on. So this idea of capturing, counting, characterizing circulating tumor cells is a really, really hot area, I guess I'll say, within bioengineering as well as cancer research. So I think I had a question from...
Yeah, so are the cancer stem cells in the primary tumor site and they detach and transfer to another clinical cancer site, or do they at some point change from a different cell into a cancer cell?
So the question is the cancer stem cell in the primary site, or does the cancer...
Does it start as something else and change into
Okay. Or does a cell break free from the primary tumor and acquire these stem cell-like properties? The answer is yes. The cancer stem cells, or what we believe to be cancer stem cells, have been found in the primary site. It's an open question as to when and where these stem cell properties might be acquired as a non-cancer stem cell breaks free and starts moving throughout the body. So open research question for sure. Yes?
What would be the implication of finding out when these stem cells activate? Like, what would be the next step in cancer research after that?
Oh, yes. So this is very exciting. So the question is, if we can find the cancer stem cell, what is the next step? Right. Okay. So if we can actually really identify what a cancer stem cell is. And honestly, the cancer research community is struggling with this right now. What molecules are expressed, what general properties are expressed by cancer stem cells, because they might not be the same for breast cancer as they are for colon cancer, as they are for head and neck cancer. So we've got a whole number of things that we have to worry about in that domain. But if we can truly identify them, then it's hoped that we can take real cancer stem cells and is there a definite molecular signature? Is there a definite genetic signature? Do we already have a drug that targets those particular factors? And if so, can we use that to kill those cancer stem cells? Because as I mentioned earlier, they're very drug resistant, they're very chemotherapy resistant, they're very radiotherapy resistant. So while we might have drugs that can already kill them, maybe those drugs can't get to the cancer stem cells. So that's one issue. Now, we may not have those drugs at all that could kill the cancer stem cells, assuming we can find them in the first place. So it's a multi-pronged issue. But there is a drug in, I'm not sure what phase of clinical development. It's targeting, I think, the Notch1 pathway, for those of you who are kind of stem cell wonks. The Notch1 pathway, pretty sure, that's coming out of Michael Clark's lab. Michael Clark is the PI who's attributed with finding the breast cancer stem cell signature. So there are efforts to target stem cells and stem cell pathways to kill cancer stem cells and ultimately the cancer itself. Sound okay? Okay. Yes, one more question?
During this process, like metastasis, are there any other mediums in which these cancer cells use to travel throughout the body other than just the bloodstream?
Yes. Yep. So there's this whole other set of research that's going on related to lymphatic metastasis. So metastasis from the primary site to lymph nodes, oftentimes through the lymphatic vasculature. And trivia, one of the leading researchers in that area is Melody Schwartz, who's from my PhD alma mater, Johns Hopkins. She is a chemical engineer. And she is really leading the charge on this. Now, there are lots of other people who are doing lymphatic research who are doing a lot of good work as well, but I wanted to just give another shout out to some engineers who are doing some top-notch work. So we have lymphatic research, lymphatic metastasis, as well as hematogenous metastasis. This is the blood system that I'm and my lab are primarily looking at. There's also more of a, how do I say this? There are other mechanisms, although for distant metastasis, we're typically thinking about hematogenous, bloodborne, or lymphatic transit. Because to go from the lung or the breast to the liver or the bone, it's a long way, and you're not going to get it by tissue-to-tissue contact. You're much more likely to need those superhighways to get from place to place. Another question? Yes.
Are there any particular biochemical or biophysical conditions that cause the transmigration from the primary site to the next site?
Okay, so the question is, are there any particular biochemical or biophysical conditions to help move this cell from the primary to the secondary? Yep. So I would say in a very general answer, yes, it is all biochemistry, it's all biophysics. Now, I think the biophysics tends to get the short end of the research stick here because the models, the tools that you need to study the biophysics of the stress on the cell, the stress on the molecules themselves, there aren't that many people who have that specialized training, and the tools that you need to study those types of interaction or those types of phenomenon are quite limited as well. I would think that I gotta give it up to Dr. Tees here. He's well versed in a lot of those different tools. Now, in the biochemical aspect of it, this is where most research is performed. I think if you broke it down to, let's say, tumor immunology, this was the subject of the Stand Up to Cancer special. All of these different new ways to harness the body's immune system to fight cancer. So that is pretty much all biochemical. But if we're talking about which ones are the most special, something like a cytokine or a chemokine versus an adhesion molecule, so a stimuli molecule versus an adhesion molecule, it's where is your training bias? I care an awful lot about those adhesion molecules, but perhaps my colleagues who do more traditional immunology research are really more concerned about what is that insult that just makes a cell angry and makes it want to kick out a therapeutic or become a stem cell or simply not die. So I'm not sure I totally answered your question, but we can talk more after. Yes?
I've got two questions. The first question is regarding the science aspects. So the stem cell research gives us a very exciting preview which can help us cure disease.
Uh-huh.
And this is positive feedback. So what do you think about the negative feedback about stem cell research?
Okay, so
So for example, if we do like, go to the other side, the opposite side, what's the danger to the human beings if we do like stem cell research?
okay, so the question was that there's a lot of research into the positives of stem cells.
Right.
What are the negative
Potential danger.
potential dangers of undertaking
Right.
research in the stem cell domain?
Right.
Okay, so that actually will lead me into the next portion of my talk, which I only have a tiny bit of time. Okay. So cancer stem cells, I think most of us agree we gotta kill them. We gotta figure out what they are. We have to target them. They're pretty nasty. But in the world of therapeutic cell research, so the world of embryonic stem cells, the world of hematopoietic stem cells, the world of induced pluripotent stem cells, there is much more controversy. Am I taking this in the right direction? Oh, okay. So before I get to that question, so embryonic stem cell research. When you hear in the news stem cell research, more often than not, there's the implication that it is embryonic stem cell research. And when we say embryonic stem cells, so I'm sorry, I'm going to draw. I don't need to draw a picture. So 4 to 5 days after fertilization of an egg, no matter what species, we'll just say, any species you'd like to talk about, a small mass of cells grows. And in this small mass of cells, called a blastocyst, for those of you who are interested, in the inner cell mass, there's about 30 out of 150 cells in this 4 to 5 day fertilized egg that are the embryonic stem cells that are used for research. So a lot of the bioethical controversy comes about as to, is this embryo, is it life or not? Thus, there are the deep bioethical conversations that are had throughout society, science, the magazine, Nature, the journal. There was a case recently that went all the way to the Supreme Court. So depending on how skeptical you are, there were a pair of researchers who sued the Department of Health and Human Services, led by Catherine Sebelius at the time, to say that the NIH needs to stop funding embryonic stem cell research. Bad. The Supreme Court threw out the case, saying, or essentially allowing the NIH, the National Institutes of Health, to continue funding embryonic stem cell research for therapeutic purposes. So again, when we hear stem cell research, we're typically thinking of this embryonic stem cell research. So yes?
Could this embryonic stem cell differentiate to bad things?
Yes. Yes. So in the world of embryonic stem cell research, there is the great hope. We're going to make a new liver. We're going to make a new heart. All of these things. In the research that's been performed to this point, embryonic stem cell world, cells are trained through the property of differentiation to become liver cells, or maybe not entirely functioning livers, but liver cells. Once these embryonic stem cells or other types of stem cells are placed into animal models, for instance, or for the con artists, these cancers called teratomas are oftentimes discovered. So you've got this great thing, a stem cell, be it an embryonic stem cell. I'm not talking about the bioethics. I'm not saying it's good or bad. I'm just saying here's an embryonic stem cell. Here is this other type of stem cell, an induced pluripotent stem cell. Here's this other type of stem cell. Once we train it to become this new liver, these pancreatic cells, whatever other cell you're interested in, we need to make sure that bad things don't happen. And to this point, for these different types of stem cells that have been trained to differentiate and become potentially new organs, oftentimes the side effect is a cancer, and the cancer is oftentimes this teratoma type cancer. So in the world of therapeutics, yeah, we think there's hope. We can make these therapeutic stem cells, so not cancer stem cells. We can make these therapeutic stem cells become practically anything we would like them to become to cure a host of diseases, to alleviate the organ transplantation need. But we haven't gotten to the point where we know that these new organs, these new cells, these new tissues that arise are going to be safe and do the job that they're supposed to do, be a new liver. So yes, another question. So are there specific conditions that can result in the formation of a stem cell? Cancer of the stem cell? Okay, so this is an interesting subject right here. So another whole area of research. So the cancer stem cell, how it's formed, I think a lot of the questions have had to do with where, when, why. Yeah, one of the open questions is whether or not a cancer stem cell is a series of mutations that happened in what otherwise used to be a regular intestinal stem cell, a regular skin stem cell that we all have, or a pancreatic stem cell. The answer is we don't know, but there's a lot of people trying to figure that out for sure. Okay, so we'll go on with the questions. If I don't need my notes, so yes.
So what exactly is the teratoma?
Yeah, it's a germline cancer. Oh, yes, I keep forgetting to do that. So what is a teratoma? So I'm pretty sure I said it correctly. Teratoma is a germline cancer, which means it is a very primitive type of cell cancer. It's not a very common one in the general population, but it's one that's frequently seen with these studies that are performed by legitimate research labs trying to figure out whether or not these therapeutic stem cells can be used. But they're also the type of cancer for those con artists that I keep mentioning, when they take stem cells, and these are stem cells in quotation marks, and put them into desperate patients. These are the types of cancers that typically arise in these people who are looking for hope, cure, and instead they get cancer. So, yeah. Other questions? One more back. Oh, in the back, yes. Oh boy. So the question is, if you're a carrier of the genes, the cancer stem cells, or a potential to maybe generate cancers with a lot of stem cells or stem cell properties, what is the likelihood of passing it on to your children? Whew. Okay. I wish I knew the answer to that question. Unfortunately, I don't. And I'm not 100% sure whether or not it's a well-defined research area right now as we struggle with what is a stem cell really? And the world of epigenetics is also really, really reaching its maturity point now that I think the question that you asked will be addressed by the research community soon, but I don't know if it's really being looked at right now. Ooh.
You made it.
Yeah. Oh, my.
I'm sure Dr. Burdick would be happy to answer additional questions if you guys want to come up afterwards, but we're kind of at the hour, so we want to be sure we dismiss those that need to go. Also, don't forget, next Tuesday is the Sigma Xi Ice Cream Social. I get the next cafe conversation, the first cafe conversation of the year will be next week, African music and drums, dancing. And then the week after that, we have our second science cafe on
Climatology.
thank you,
Climate
climatology.
change.
I remember. Climate modeling, climatology. So all things you should keep on your calendar. All of them are at 5 o'clock here, except the ice cream social. It's outside. 5 o'clock still. Time's the same. All right. Thank you very much, guys. Glad you came.